Yes, you can start now.
Okay, sir. Good afternoon to one and all present for today's session. Myself, Megha Hundekar, PhD research scholar from Mysore University. Every Sunday, we are trying to explore the diversified facts and the corners of life sciences. I would not say specifically microbiology, but the different corners and aspects of life sciences. Every Sunday, we are trying to make aware all the microbiologists and all the life science members about what are the things going in and around. So today is yet another very wonderful session, and that is nothing but open discussion on chemolithothrophs. So today we are having with us Dr. Nitin Adapure sir, who is very well known for his work in the chemolithothrophs. So it's my privilege to welcome Dr. Nitin Adapure sir for today's session. We welcome you, sir.
Yes, thank you.
Even I would like to welcome all our members today for the session. Today we are going to have an open discussion on chemolithothrophs, where Nitin sir is going to first highlight on what is his research on chemolithothrophs, and also try to highlight on few aspects which needs to be covered in the future aspects. So over to you, sir.
Thank you. First of all, I would like to thank Microbiology Society for arranging the talk on, or we can say the discussion on this wonderful topic, that is the chemolithotrophs. Obviously, all the attendees they are all microbiology or biotechnology students, so they definitely might have studied about the chemolithotrophs, and some of them they might be working also on this particular topic. So like that, the chemolithotrophs, this is actually a very large area, I'll say, and among that, on one of the type of chemolithotrophs I have actually worked, and actually still I'm working on it. Let us first of all start with basically that what is mean by chemolithotrophs. So definitely all of you are aware that what is chemolithotroph, but as a part of your we can say starting the discussion, I would like to say that what is chemolithotroph. So you might have studied, all of you have studied the nutritional classification of microorganisms. So among that we have come to know there are certain, basically any organism, it requires energy, it requires carbon, and it requires some electron donor. So on the basis of three aspects, the categorization of microorganism has been done. So on the basis of getting energy from any component, so there are two categories, as you know, that these are the, on the basis of energy, we can classify the organisms as the phototrophs and the chemotrophs. So phototrophs and the chemotrophs. So the one which is getting energy from the sunlight, they are called as the phototrophs, and the one which is getting energy from the chemical oxidation, or we can say the oxidation reduction reactions of the chemicals, so these are called as chemotrophs. Now the second type is on the basis of carbon source, the organisms which are getting carbon from organic components, so they are called as organotrophs. And the one which is getting carbon, so I'll say that chemolithotrophs, if you are talking about, so the one which is taking carbon from the organic component, this is a one category, and the one which is taking carbon from the CO2, that is CO2 is a carbon source. So these are the autotrophs and the heterotrophs. So the one which is getting carbon from the CO2 is autotroph, and the another one which is getting from the carbon from the organic component, so they are the heterotrophs. And depending on the electron donor, now there is a categorization called as litho and the organotroph. So here, if the organic component is electron donor, so it is an organotroph, and if the inorganic component is electron donor, then it is a lithotroph. So if we have to categorize it properly, so like for today's session, I would suggest the title would be chemolitho autotroph. This would be the title. More appropriate one, because the chemo, litho, and autotroph, so on the basis of three categories we have categorized the organism. And so like that, if we can take the examples, so taken typical examples of E. coli or Salmonella. So the routinely used organisms in our laboratory. So they are categorized as chemoheterotrophs, chemoorganoheterotrophs. And generally we can say chemolithoautotrophic organisms are not routinely cultivated in we can say in day-to-day practicals or in typically in laboratories. So the one who is specifically involved in such kind of research, so only these students or researchers they are actually cultivating such kind of organisms. Otherwise, yeah, though it is a part of some practicals in some universities, but people face some problems in cultivation of such kind of organisms. That's what actually I have observed around. Talking about my research work. So as said by Megha ma'am, like among the chemolithotrophs, I have worked on iron oxidizing organisms. Yes, actually, I think I have missed one point. Like if whenever we are talking about chemolithotrophs, so obviously CO2 is a carbon source. Then inorganic compound, they are energy source, and chemicals, the chemical oxidation reduction reactions. These are the energy source. So they can be categorized into various types, or they can be you might have heard about iron oxidizing organisms, sulfur oxidizing organisms, ammonia oxidizing organisms, nitrite oxidizing organisms, so even hydrogen oxidizing organisms. So these are the typical examples, or we can say typical we can say subcategories in chemolithotrophic organisms. So it is a part of syllabus in some universities that the physiology, the electron transport chain is being taught. And so here the electron donor, as we have discussed, is the inorganic one. That is in case of iron oxidizing, it is iron, Fe2. In case of the ammonia oxidizing, it is ammonia. So in case of sulfur oxidizing, it is sulfur. So this is what we can say the electron donor. And the electron acceptor, it varies. Like in typically the electron acceptor is oxygen. So that's why these organisms, they are aerobic chemolithotrophic organisms. Yes, but there are certain anaerobic chemolithotrophic organisms existing in nature. So actually it is a bit tricky to cultivate such kind of organisms and to identify its growth. But with practice, one can easily habituate with cultivation of such kind of organisms. Uh, so in my work, I have handled iron oxidizing organisms. So these are you might have heard about Thiobacillus peroxidans. So the we can say recent name is Acidithiobacillus peroxidans, and even there is existence of Thiobacillus caldus. Then what we can say Leptospirillum. So these are the typical organisms which are involved in iron oxidation, and the communities involved in iron oxidation. So I have enriched these organisms from mining samples. So I have taken the mining samples, pyrite mine samples I have taken, the bauxite mine samples I have taken, and then I have used these samples for the cultivation of this iron oxidation particularly. So later on, I have used these organisms for leaching out the metals from the electronic waste. And so currently we have actually in long back in 2008 we have started with this work with the successful studies of the laboratory scale. Then later on, for the pilot scale studies of this metal leaching, we have got a funding from the BIRAC. And right now we have a 10-liter as well as 100-liter scale studies for or we can say demonstration plant for this electronic waste, the metal leaching from the electronic waste, by using this iron oxidizing organisms. So I am currently using these chemolithotrophic organisms, particularly iron oxidizing organisms, for the metal leaching from electronic waste at 100-liter scale. And this TRL level is right now it is 7. Technology readiness level is 7 right now, and hopefully very soon it will be get commercialized with the help of BIRAC soon. So this is what we can say the use of chemolithotrophic organisms in my laboratory. I am doing this. So now I think this we can say that this much of introduction is sufficient, I think. And let's open the session for discussion. And as I said, this is a very large area, chemolithoautotrophic organisms. Among that, I have used only one type of organisms in my studies. And so definitely, whatever the little bit things I'm knowing about this nature's creation, that is the iron oxidizing organisms. So I can surely, tell you or guide you about such kind of organisms. And let's start the discussion. So yes, anyone can ask question. I think, yeah, whatever you like, one can raise your hand or can ask the questions directly. Megha, is it okay?
Yes, sir. Yes, sir. Or anyone those who are having the questions can unmute yourself, or if not possible, you can text your questions in the chat box.
Yes, you can place your questions in chat box as well. So either way, what is feasible for you, so you can go with that.
Yes, and one more thing.
Yeah.
If
Yes.
anyone is working at present on chemolithotroph, they can mention the topic of their research, topic of their dissertation, and also can make a comment. Anyone.
So my question for you is, Nitin sir.
Yes, yes.
So as you said that cultivation of this chemolithotrophs is difficult in the laboratory. At present, do we have any ready-made media that can be used for the initial screening and cultivation of this chemolithotrophs in the lab scale?
Yes. So as I said, I will talk particularly on iron oxidizing organisms. Initially, we'll discuss about it. Let us consider that if one has to grow the organisms, the iron oxidizers one particularly, so one has to take the samples, collect the samples from the mining site. Say pyrite, typically pyrite mines, they are more abundant with these organisms, iron oxidizing organisms. So you have to take the samples, you have to crush the ore, and then you have to prepare a media. The media generally used for cultivation of these organisms is 9K medium. 9K is 1,000. So yes, so this media is suitable for the cultivation of iron oxidizers. The composition is readily available, and components, they are also easily available. Ferrous sulfate is the main component because this is the main energy source for the organism. As I said, the name itself is suggests iron oxidizers. So Fe2 is an electron donor. So that's why it needs an abundant amount because this is the main energy source. And there is no any as such ready-made media available, though some media suppliers, they might be supplying this, but I don't think there is a necessity of taking the media from such kind of suppliers, because the compounds are readily available. If at all, if you need, you can take it. But we can say it is having general medium composition. Ammonium sulfate is there, then magnesium sulfate is there, potassium chloride is there, and ferrous sulfate is there. Traces of calcium nitrate. So this is typically available in every lab. So you can prepare the media and go with the cultivation.
Sure, sir. Sure. Thank you, sir.
This is, I'm talking about the liquid media. I'm saying, well,
Yes,
you have to enrich the organisms in liquid media first. So here you have to use like the traditionally what we are doing. We are taking the samples and then we are going with the plating and plate preparation, or solid media we are going. So in this case of iron oxidizing organisms, we should go first with the enrichment. So after the enrichment, you can go with the plating.
Okay.
Yeah.
Can you focus some light on maintenance and transfer of this culture?
Yes. So as I said,
what is the survival period of these cultures if we try to maintain in the refrigerator?
Yes. So in this case, again, we can say that traditionally, whatever the things we are doing in the typical microbiology laboratory. So in this case of chemolithoautotrophic organisms, the things are a bit different. Here, if you have to store the culture, so in liquid form, it can be stored for say months, 6 months, or something like that in a liquid form. as we can say the enriched flask if you have. So that flask you can keep it for 6 months or something. But again, we can say whatever the time of storage it increases, so again it will take much higher time for the, we can say for the re-enrichment, or we can say for recultivation or activation, I'll say. So this is what actually the things happen. So if you need activated culture, you go on frequently subculturing it. Subculturing is nothing but taking a fresh media and go on subculturing, so that the iron oxidation rate it is maintained. Because see, in this case iron oxidation rate is the important factor, because if we have a faster iron oxidation rate, that is in minimum time it can oxidize complete iron. So this is the such kind of organisms, they are actually more having a more value in terms of application point of view. No doubt the organism it will survive, because see, in nature, in a natural habitat, they are residing on the ores. So on ores there is no any ample amount of water available. So in we can say in a dried, little bit dried form, it is existing in nature. So if you want to store it, even you can store it in a ore form only. Like you have ore sample, you can you have enriched the samples, then the remaining part of the ore you can keep it as it is. Or else what you can do is you can again add your culture in that ore sample for the preservation purpose, so that whenever next time you have to you are in need of these organisms, you can take the same ore samples and you can you can enrich these organisms. Because ores, these are the natural habitat of these organisms. So you are keeping the organisms in its natural habitat. So this is actually the better thing.
Very nice. You have explained it nicely.
Yeah.
Then again, I will request others. Shashi Chawla sir is there. Please, if your student have done sometimes the work of chemolithotroph, Ketan Narkade is there, Nagrani ma'am is there. So you can focus some light. Kohima. Kohima is from which place? Kohima. Gupta, Shraddha Gupta.
Excuse me, sir.
Yes, Gauri, please.
Yes. Hello, everyone. My name is Gauri Bhavkar. I am Dr. Shivaji Wagmare's PhD student from Ferguson College, Pune. Uh, First of all, this is a very interesting, meeting where I've always been referring to Dr. Adhapure's works actually. So I made it a point not to miss this opportunity to have a discussion with everyone. Dr. Adhapure is going to guide us about chemolithothrophs. Actually, I've just started with my doctoral research work. Even I am trying to work in the area of electronic waste management, and as I started referencing, I came across Dr. Adhapure's work actually. And I have done sampling from copper mines in the Malanchkhan area now recently, and I have been trying to isolate iron oxidizing bacteria, and just in the enrichment phase nowadays. Okay, so according to the referencing book, I've also been using this 9K medium. But I have not been able to, have any success with cultivating them on solid media. And I've tried using, double strength medium, and then, autoclaving agar separately and then mixing it and everything. But as far as whatever work of 2 months that I've started, I've not been able to successfully cultivate them on solid medium. So I would request Dr. Adhapure to shed some light on this area. If it is possible to how is it possible to cultivate these organisms on solid medium?
Yes. So as I said, initially, whosoever is working on these kind of organisms, so first of all, in my opinion, first of all, should go with the enrichment first. If you are enough confident about its enrichment, that yes, it is quite appropriately enriched, so then you can go with the solid medium. Now, in my case, again, I would suggest why there is a need of going with the solid medium. Like, what is your final purpose? Like, do you need any, if the purpose is just to go with the isolation and identification, that's it. In that case, yes, you can go with the solid medium. But if if the purpose is like application, so in that case, even if you have the mixed consortium, whatever the consortium you have got in liquid form, so that consortium you can use it directly for application. It doesn't matter that if it has a single organism or multiple organisms, because the main purpose of leaching or the application, whatever the application you are doing, if that purpose is getting solved, so then I don't think there is any need of go with the isolation. But yes, as I said earlier, like, if at all, if the person is interested in microbial diversity studies, in that case one has to go with the identification and getting a pure isolates. So in that case, you can definitely go with the solid medium. And for that, actually, I have mentioned the protocol in my previous publications. There is again here the things are different as compared to the chemoheterotrophic organisms. Here, since the pH is almost 2 or below that, so in that case, the agar-agar itself, it gets degraded during the autoclaving. We can say acid hydrolysis. It takes place of the agar-agar, and so agar, it gets broken down. So in this case, we have to take the agarose particularly, or even one can use gelrite as well. So this is taken in another aliquot, and with a simple distilled water. Then your ferrous sulfate is taken in a concentrated form, and then it is added after we can say autoclaving, because again ferrous sulfate during autoclaving it gets hampered, and the remaining media components, they are sterilized by autoclaving. The ferrous sulfate is sterilized by filter sterilization. So this is how the media is done, media is made, and later you can go with the media plating, typically as we do it with the chemoheterotrophic one. So definitely you'll get the isolates. Fantastic, very beautiful colonies are obtained. We can say rusty colored or dark brown colored colonies are obtained. So these are nothing but the isolates, the particularly isolates of the iron oxidizing organisms. So this is how the isolation is done in this case. So the methodology is well mentioned in we can say in a very in so many research articles, even in my research articles as well. So you can go through that. It is not that much difficult.
Thank you, sir. I shall be search for your more reference work. And I have not tried with agarose or gelrite, but I have tried the same procedure with agar, double strength agar actually. So for spread plating it worked, but then for streaking it didn't work. I'll have to give it a try again. And whatever color, I got yellow colored growth on the plates. And what happened was an uninoculated plate that was also incubated with the other plates, with the incubator. That also changed its color actually. That was my reason for worry. But let's see, I'll have to discover, I'll have to explore more. I shall definitely try to search for your reference work, sir. Yes, that acid hydrolysis thing and that filter sterilization thing, I have experienced this, that the ferrous sulfate changes its color from green to yellow directly after autoclaving. So filter sterilization is essential there. That is right. And essentially when the medium becomes very light green in color, it is a technique which is which can easily be mastered now. That is true. This work is quite interesting. Working with chemolithothrophs is very, very interesting and very different from routine cultivation of bacteria. And even I am interested in isolation and identification of organisms. So that's why I want to go for isolating organisms on solid media actually.
Okay, there is Megha, there is one more question in chat box.
Yes, sir. It's from Kohima. With this study, can we relevantly focus on starving cancerous cells isolate? I keenly want to know about anaerobic glycolysis, reactive oxygen species, oxidative stress, and defective mitochondria metabolic diseases. So she's willing to know how can we correlate this with starving cancerous cells, sir.
Actually, in my opinion, this is not actually related with the cancerous cells, or there is no any relation with this, because here in chemolithotrophic organisms, we are talking about the organisms which are using inorganic component as an energy source, particularly. So as I said, key iron oxidizers, sulfur oxidizers, then ammonia oxidizers. So like that, these organisms, they are involved. So I don't think so. Is there any relation between the reactive oxygen species, anaerobic? See, these are different things which she has mentioned: anaerobic glycolysis, reactive oxygen species, and oxidative stress. So
Yes,
personally,
yes, you
I
are
don't.
right. You
Yes,
are right. You are.
I don't think there is any relation between chemolithotroph and the topics which she has mentioned.
Yes, sir.
Yeah.
So there is one more question. Can you please highlight on the industrial applications of chemolithotrophs? It's from Amrish sir, from Dharwad.
Yes. So particularly, I will say right now, let's start a discussion with iron oxidizers itself. So as I said, we are using it for like metal leaching, bio leaching. You have heard. So bio leaching of metals from electronic waste. That's what the application which we are using. Apart from that, we can use it for desulfurization, like the same organisms or sulfur oxidizing organisms used for desulfurization of coal. So you might be knowing that in coal industries or coal mining, so there is a problem of sulfur. So that desulfurization can also be can also be facilitated by using these organisms, chemolithotrophic organisms. Then, as I said, there are certain ores from which the metal leaching is not feasible, like low grade ores. So as we know, typically from the mining side, how the metals are recovered from mines. So generally the physical processes, smelting and all, this is done. But this is done with the high grade ores. The ores which are having a higher percentage of metals, they can be used for smelting and other purposes, and the metals can be recovered. But what happens with the low grade ores? The ore which are having a very less concentration of metals. So in this case, the bioleaching can be used. Like we can, we can use microorganisms. We can exploit the microorganisms for the recovery of metals from this low grade ores. So this is an application for metal leaching. Then again, I would say, let's talk about ammonia oxidizers. So I would be happy to tell you that one of the consultant and working in microbiology area, so he has visited our laboratory, and actually in our lab we are actually cultivating various physiologically different kind of organisms in our lab. So the photolithotrophic organisms, then what we can say, anoxygenic phototrophs, oxygenic phototrophs. So different kind of organisms we are growing in our lab. So he has visited the lab and he has asked me one question: do you have ammonia oxidizing organisms with you? I said, right now I don't have. So he says that if you have it by any means, or if you get it by any means, so tell me immediately. I am in urgent need of ammonia oxidizing organisms. I ask why you are in need of that. So he says that there is an industry in South something, Chennai or something. So they are having a large ammonia content in their effluent. So they have used different technologies for removal of ammonia from it, but that they are not that much successful. So I want to use this ammonia oxidizers in that, so that since ammonia is the main energy source, it needs an ample amount by the organisms, and so in this way the purpose is solved. So like that, there is a great demand of ammonia oxidizers as well. Like 1 or 2 years back, we have actually cultivated such kind of organisms in our lab as well. But again, as I said, it is very tricky to, number one, isolate the organisms or enrich the organisms, and number two, to preserve them. So actually, though we have successfully isolated the ammonia oxidizers or enriched the ammonia oxidizers, but later on, actually, we failed to enrich it. Actually, we didn't get a time to focus on the enrichment of this kind of organisms. But what I'm saying is, as the question has been asked, what is the scope? So yes, the influence which are having a higher concentration of ammonia oxidizers can be depleted over there. Like that, the nitrite oxidizers, yes, if there is a problem of nitrite somewhere, anywhere, like so you can deploy these organisms. Even we can say hydrogen oxidizing organisms, they are there. Carbon monoxide oxidizing organisms, they are there. So now we can simply imagine if one has the carbon monoxide oxidizing organisms. So we are talking about, you know, the pollution of caused by the carbon monoxide. So if one can have organisms which are growing on them, so simply we can exploit them. So like that, the applications are, you know, n number of applications they are there, but we just have to deploy it right organism at the right place. So that's what we can say the work of a human is, because nature has provided, you know, large number of organisms with a greater variety in the nature, but the only thing is it is a human failure that we are not identifying the right organisms and for the right application. Yes, Megha, please.
Truly said, sir. Sir, just like you mentioned that this chemolithotrophs or chemolithoautotrophs, they are used usually for bioleaching of metals. Can we have the same organism for production of certain enzymes that are also industrially important? Apart from bioleaching, can they be used for general production of some enzymes which have different industrial applications? Like usually we say that all the enzymes are produced by regular microorganisms which have a normal optimum parameters. So can we make use of such kind of chemolithotrophs for a production of any kind of enzymes that are industrially important?
Yes. So that's again since the organisms, they have a different physiology. So like that, we couldn't get a, say, amylase producing organisms. Just for example, I'm saying if we are looking for amylase producing organisms in chemolithotrophs, so certainly we would not get it. But definitely they have some different kinds of enzymes which can be which can be used instead of using the pure organisms. See, for example, like as I said, the growing such kind of organisms and maintaining or preserving them, it is a bit tricky. So in that case, what we can do is, once we have a ample amount of cultivation, we can extract the enzymes, okay, and we can preserving the enzymes. It is quite simpler. So we can preserve the enzymes, and then we can use it later on for the same purpose, for which we are using the microorganisms. Even we can have the immobilization option as well. So like that, we have to go on using the different we can say different options for utilizing the particular enzyme, again the right enzyme at the right place. So like the rusty cyanine is the enzyme in present in this iron oxidizing organisms. So again, there are certain studies for extracting the rusty cyanine and using it for, you know, kind of application. So like that, we just have to identify like which enzyme can be used, which enzyme can be first of all extracted, and then it can be used for this kind of purpose. So, but certainly, yes, as we are doing it with the chemoheterotrophs, we can do it with the chemolithotrophs as well.
Okay, sir. Thank you. Members, any more questions? Anyone wants to raise any question? The session is open.
Yes, again, like Megha is also requesting all to ask the questions. I would also request you all that you can ask your queries or questions. Definitely, it will be helpful for, you know, for others and for me too.
Correct. There was a good discussion, but anyone working on chemolithotroph having any difficulty on chemolithotroph can ask the question or can share the experience, not asking the question.
Yes,
they can share their experience also.
Yes.
Okay.
Yes, it may happen that some of you they are working not only on iron oxidizers, but on other types of chemolithrophs. It may happen. So please share your views. It will be helpful for all.
And if not, we'll conclude the session. No problem.
Yes, sure.
Yes, and there was really a good discussion. Again, I would like to tell you, very few teachers and scientists in India are working with the chemolithotroph. The reason is again the same: difficulty in isolation. most of the student wants a PhD topic should be simple and I should complete it within a short time. So they never dare to touch to isolation or any application of chemolithotroph. So the work research on chemolithotroph has tremendous scope and tremendous research is waiting in this field. That's why we have arranged a group discussion on chemolithotroph, or better to say chemolithoautotroph. So Tamanna, sorry, Megha,
Yes, sir.
you can conclude if there is no question, if no one is raising the hand, then you can conclude.
Sure, sir, sure.
Yeah,
So my
before
last
that,
question to Nitin, sir. So
okay.
just like you said, there are many opportunities to work in chemolithotropes. If you can just put some limelight on a few small topics that can be given for dissertations to students, or even as topics for your for the research scholars. Because what I have felt through this journey with the research scholars group is many people are just running behind nanotechnology, they are running behind bioplastics, we are running behind enzymology, we are running behind AMR, but there is no more interest shown in this arena of microbiology, that is chemolithotropes. So if you can just give me 2-3 glimpses of what can be the topics for a research scholar or even for students from the colleges for the dissertation, small projects or even as a topic for the research scholars.
Okay, fine. In that way, actually, I just want to not only on the topics, but also, can I share the screen actually?
Yes, sir. Yes, sir. I'll just make you the host. Just a minute. Yes, sir. You can share your screen. You're the co-host now.
Okay, fine, fine. Like, I hope this is visible to you all. And yes, is it? The screen is visible to you?
It's visible.
Yes, sir.
Yeah, sure. Okay, so so as I said, there are so many different types of autotrophic organisms existing on Earth. So these are the few examples. Again, I'll say, and so as I said, this iron oxidizing organisms here, as mentioned here, diabacillus peroxidans I have used, but this is not the only organism. So there are so many different types. Like as I said, carbon monoxide oxidizing bacteria. So example is Pseudomonas carboxydovorans. Ammonia oxidizers, nitride oxidizers, acetogenic organisms. So they are producing acetic acid. Methanogenic organisms. So here we can say that like hydrogen is the electron donor. Like in case of iron oxidizers, iron is electron donor. And as I said, whenever you have to grow a particular type of chemolithotroph, say here example is iron oxidizers, you have to provide iron in an ample amount. Like say in 100 ml of media, you are giving almost 4.5 grams of the ferrous sulfate. So in such a higher amount, you are actually adding the energy source. So like that, in case of this, the methanogenic, it is a hydrogen is the electron donor. Acetogenic, again, the hydrogen is electron donor, but the product is different. So like that, you can use different organisms. And again, we can say this could be the topic. Like whatever the applications we are actually the organisms we are actually discussing, so these the same could be the topic for your work. But as I said, generally what happens is your dissertation, say for example UG or PG dissertations, so they have a limited time of say 6 months or something. So
Yes, sir.
growing such organisms in a limited time, it is bit we can say risky kind of a thing, because if you didn't get a results, so you don't have anything to write in your dissertation report, because what students or what the teachers or even what the examiner is expecting is a typical thesis, complete 50 to 60 pages thesis. and
sir.
they don't believe that you have tried to grow such kind of difficult organisms and you have failed to grow it. They will not go give kind of a weightage to this. So what I'll suggest is, or what we also do typically in our lab or even in our curriculum, is we give two topics to the students. One is this kind of a topic, which is, you know, a kind of a tricky and kind of a risky kind of a topic, and simultaneously we also give a simple topic, so that, you know, at least if he fails to do the isolation or enrichment in this topic, so he has something in his hand, so that that can be used for their dissertation work. So that can be done. And what could be the we can say applications. So as I said, carbon monoxide, we can, if you are claiming, if you have a kind of a device which can be, you know, which can be kept in certain places of high polluted areas of a city, say for example, or in a company, so there definitely these organisms, if you are putting these organisms, so definitely they are actually minimizing the level of CO, if you are talking about carbon monoxide oxidizing bacteria. So like that, it can be used with we can say the other organisms. And I also yes, can I, okay, yes, now there is another, we can say diagram in front of you. So this is a redox cycling, or these are the typical diagrams from, you know, Brock biology of microorganisms. You can easily get it. So what I want, why I have purposefully shown this particular diagram to you is, so this is what happens in nature: ferrous to ferric, ferric to ferrous, if you are talking about iron. Okay, so this is naturally happening. Some of the organisms, they are utilizing ferrous and converting into ferric. This comes under chemolithotroph. But if you are talking about conversion of ferric to the ferrous, this is the reduction. These are the anaerobic, this is anaerobic process. So we can say, or even in case of leaching, this is aerobic as well. So what I'm saying is, all these things, they are happening naturally. So what we have to do is, so in nature, all types of organisms, they are existing. Here I'm showing about the sulfur. I hope you can see this sulfur cycle as well. Is it, Megha?
Yes, sir. Yes, sir. It's
Okay,
visible.
so here also I just want to point out that there are certain H2S oxidizing organisms. So this will come under chemolithotroph, but surely this sulfate can also be, you know, converted into H2S. So this is sulfate reducing organisms. So here, though we are talking about chemolithotrophs, but whenever we are discussing about topics for dissertation, one can point out on this sulfate reducer as well. So like once you are trying to grow such kind of, we are having this sulfate reducers in our lab even. So we have cultivated these organisms. You know, the black color of this wastewater, it is because of this sulfate reducers. So it is producing H2S. So sulfides, they are also being produced. So there are different types of organisms existing in nature, and they are converting one component to the other component. The mechanism is different. We have categorized this mechanism in under different headings. But if we are looking as a, you know, from microphysiological point of view, so see such a greater we can say variation is there, which is existing in nature among the microorganisms. So we have to use that. We have to exploit these kind of organisms. And yes, whatever the way you are actually choosing to exploit it, so that will be your project. That's what I just want to tell.
Yes, sir. Yes, sir. Truly, sir. Truly, sir. So there are two more questions from Kohima. One is, what is the energy absorption mechanism for chemolithotrophs and mechanism of ATP production?
Yes, what is a?
What is the energy absorption mechanism for chemolithotrophs?
Energy absorption mechanism. See,
Yes.
if you're talking about iron, so like iron oxidizers, so yes, Fe2 is giving electron. So that electron is taken up by the one of the cytochrome in the membrane, and then it is the electron is transferred to the rusticinin and then to the electron transport chain carriers. The electron transport chain carriers, it varies from organism to organisms. Whenever we are talking about iron oxidizers or sulfur oxidizers, so these carriers, they are varying. And as we know that the typical process of ETP, that is so ETC, electron. So as I said, there are since the organism, it varies, iron oxidizers, sulfur oxidizers. So electron transport chain carriers, it varies. And the typical mechanism, as we know, electron is carried from one carrier to the other carrier. And during this time, the protons are getting pumped to the periplasmic space. And then what the protons, they can come back by ATP synthase. That's what we know typically about the chemoheterotrophs as well. So the same thing, it happens. With this, the only thing which differs is we can say the electron donor is different. Like in chemoheterotroph, NADH is electron donor, like in E. coli or even in humans as well. So NADH is the electron donor. But in this case, the electron donor is different. So this is what we can see the difference is. And the ATP is synthesized typically by, we can say ATP synthase, through the ATP synthase. Now here the NADH is again regenerated by the reverse electron flow. And this is how we can say the NADH is generated.
Okay, sir. Thank you, sir. Deshmukh sir, can we conclude the session?
Yes.
Okay, sir. So today it was really a very wonderful session. I could say that I got some more knowledge about the unknown arena of microbiology, that is the chemolithotrophs. As even I am a research scholar, I was not much aware of this chemolithotrophs. So I would like to thank Dr. Nitin sir for giving his valuable time on Sunday, though it's a holiday for all of us, for sparing his time and giving us information about his research work and what can be still done to cover the arena of chemolithotrophs. Thank you, sir.
Yes, thank you. Once again, I would like to thank Microbiology Society for arranging such a different topic for the discussion, and thank you all for attending the session. Your attendance, it suggests or it indicates that yes, definitely you people having an interest in microbiology and such kind of different subjects. Thank you for.
Yes, sir. Yes, sir. Even I would like to thank Dr. Deshmukh sir for always encouraging, giving up new ideas to bring all the life science members active back to state, so that we can create a very aware society about the life sciences. Thank you, sir. And last but not the least, I would also like to thank all the members today who have joined for the session and taken some or the other information from the session and spared their time on Sunday. Thank you to all. So I conclude that the session is done. Thank you, Nitin sir.
Thank you, Adapure sir. Thank you, Deshmukh sir.
Okay. Now, Megha,
Yes, sir.
you can end the meeting.
Sure, sir. Sure. Thank you, sir. Thank you, Nitin sir.
Okay, thank you.